So I'm going to go very quickly through these slides because you've heard about it before. You know, everyone. TDCS is essentially electrodes on the head connected to a 9-volt battery, right? It's certainly not that simple, but it's a starting point to try to understand it. One electrode is connected to the positive terminal, let's say the red, and one electrode is connected to the negative terminal. And as most of you probably are aware, there's a lot of hype about this right now, right? If you look up TDCS, you're going to get more popular press articles, some of them pretty fantastic, right? That only then loosely refer to the clinical trials that were run, and everyone's thinking this stuff can make you smarter. Even more interesting is what's shown here, which is this is just in PubMed. These are peer-reviewed trials in humans using TDCS since 2000, right? We have almost, in 2011, it's probably closer to 150 papers. In 2012, I think there'll be 200, maybe 300 clinical trials with TDCS, right? So these were to be published next year. They had to be run this year. So 10 years from sort of the establishment of it, you have something that I don't know if this has happened before in medicine, and the indications are as broad as you can imagine, right? It really also, by the way, spans the extreme of age, right? So from stuff you do in an elderly to stuff you do in 5-year-olds, right? And so I was talking about whether you should be adjusting dose based on this. And, of course, looking at this, some people might say this sounds a little bit like snake oil, right? Because how can one thing be good for everything? It's just not possible, right? And so those are sort of questions that indirectly you've already heard about, and I'll be trying to speak about. How can something so simple, how can a 9-volt battery make you smarter, make you read faster, right? Help your kid focus on his math problems, and take care of his epilepsy, right? And this, and if you do on yours, and so on. So how could that be? And should we really be thinking about TDCS as one thing, right? Just slap the pads on, or is there a lot of flexibility which we're only starting to take advantage of? And the second question really goes to the question you asked about the cellular stuff, and I'm only going to be able to sort of touch on that. And I'll try. Hopefully, by the end of this talk, you'll think that okay, this crazy engineer and the engineers he works with has something valuable to tell us as far as the decisions we're going to be making tomorrow. This is really sort of the most important idea, which is one of those things that's obvious in retrospect. Obviously, when you give a drug to someone, no one assumes that the concentration of what's in the drug is what is going to appear in the hippocampus. It's not, no one's going to assume that at all, right? And no one will assume that, if you give the same drug to 2 people, you'll have the exact same concentration in the CA1 region of the hippocampus, right? It doesn't work that way. So there's the dose of what you apply, and then there is the concentration of what is generated in the head. The same thing applies for electrical stimulation. It is not the case that what you put on the outside is what happens on the inside. Yes, you control what's on the outside. You control 1 milliamp and a 3 by 5 centimeter squared pad, but you do not, whatever you want to say, that is not what shows up at the brain, right? In the brain, 1 milliamp does not show up in a 3 by 5 square meter pad, right? There's clearly a relationship between the two. And so everything I've shown you so far, all those pretty pictures, relate the two. They basically tell you, based on what you put on the outside, what happens on the inside. And obviously, if you put different things on the outside, right, different things show up on the inside. But it's not always trivial, right? The relationship between the two is not always trivial. And how do we make that relationship? We use something called forward modeling, and essentially what's done is the head is divided up into compartments, skull, CSF, and so on. And this is fed into a fancy computer program that basically simulates brain stimulation. It simulates where the current is flowing through the head. And I don't want to, in the interest of time, I don't want to spend too much time on these. Yeah.
So when you say simulation, is it pure simulation or is there any regression? In other words, are you taking it to some kind of output vector?
You're going to be optimizing it somehow?
Yeah. Do you? Again, some kind of response for any kind of response. Literally what these models are showing you to the first level is they're just predicting where current flows using a fancy version of Ohm's law.
So we literally model the head as a series of resistors. We apply the battery on the head, and we track where the current goes through. And so that's the first outcome. Once you have that information, you can do a lot more with it, right? You can.
So how do you do model selection? Is some kind of fitting parameter to measure goodness of fit? Or in other words, how do you know your models?
How do you know the models are any good? Is that your question essentially? So that's sort of what we do all day. We try to make the best models possible. So first of all, you start with very high quality MRI scans. You can always be better, but at least 1 millimeter cube resolution. Then you use those to make masks, and these masks are pretty well. The masks are as precise as the MRI, right? So if the MRI is at 1 millimeter, the masks can come out at 1 millimeter. Again, you can employ other post-processing, and you know that eye should be sort of round and CSF should be continuous. So you try to make the best masks possible, and then the actual simulation is done using finite element methods. So you generate essentially, you can see just massive meshes. So one reason, so the models are as accurate as the first approximation as the masks. And a good way to just gauge that is just by looking at it. I mean, the fact that a kid looks like a kid and I look like me, and we can even zoom in closer and look at the architecture of our brains, tells me that we're representing the brain relatively well. The physics of it is basically Ohm's law, right? Or it's basically Ohm's law in 3 dimensions. So that is a pretty sound idea. The models are used to make predictions, and I'll be showing you some examples where there were subsequent clinical trials that validated the model predictions. So I tend to think that the pictures I'm showing you are, the more time goes by, the more convinced I am that they're pretty accurate, but they're just predictions. And I'll show you more examples of that. Yeah.
Finite element methods are obviously the standard of nonlinear modeling, but you still don't know.
Linear in this case, linear.
But you still don't know, still don't have an error vector. You don't know how it fits to some kind of, how well it estimates actual reality.
And I'll show you examples of that, right. So as I said here, the models are simply used to make predictions. Then we collaborate with our clinical colleagues to feed them their predictions. They run a trial, and we'll see how well the models perform. And that really is the ultimate test of the model. And I'll be showing examples of that. It is much easier to publish a modeling paper in an engineering journal than to make something that's actually useful to clinicians. And so we try to hold even though we work on that one level, we certainly try to hold ourselves to the higher bar. So this is again what I'm showing you. This is an example of the segmentation of my head. If it wants to load up, it's maybe thinking about it. All right, maybe not. Try this one. All right, so in this case, you're not seeing 2 electrodes. What you're seeing are what look like 64 smaller electrodes. These are called high-definition electrodes. I'll be coming to it. But again, this shows you the level of accuracy that can be captured in these models. I mean, you definitely should recognize the person being modeled, at a very gross level. And you can just see that you're really talking about sort of gyri level resolution, right? So we're asking questions on a gyro, going from, let's say, this wall of the motor strip to this wall of somatosensory cortex. And by the way, you can also drill deeper into the brain. You can map current as it flows through white matter. Should we be thinking about white matter? Should we be thinking about deeper brain structures and so on? Now, most of what I'll be showing you, though, is on the surface of the head, right? Because people tend to be particularly interested in cortex. And, again, we're sitting there, we're engineers, we're sitting there with our little computers, right? So we can play around with stuff. And what we can do, for example, very quickly is screen new potential new potential configurations. Whereas in the clinic, each thing would cost you, 6 months to evaluate. We can very quickly screen them for what that's worth. We can try to think about mechanisms, and of course there's no risk because it's just a computer simulation. So here's one example. So in this case, the montage is an anode, which is in red, on the left side, and this is again sort of me, right? An anode on the left side and a cathode on the right side. This was used by a group at the University of Pennsylvania. But the general idea is that you put the anode on the left and the cathode on the right. And this plot here shows you where the current is going. So blue is weak, and yellow means that there's more current in that part of the cortex. And it's not a shocker, right? If I put the electrodes here, right, current's going to flow in through one side, and it's going to exit on the other side, so you do expect that the regions under the electrode, right, will get more juice. So in this case, things seem to be sort of intuitive. On top of that, you can ask the question, which way is it flowing? Right? So it turns out that under the anode, current is entering the head, so it actually crosses into the cortex. And then on the other side, right, it's being pulled out of the head, so it exits the cortex. So what's shown here, red shows you regions of inward current, and blue shows you regions of outward current. So under the anode, current is flowing into the brain, and in the cathode, current is flowing out. And it's believed that it's not the same thing. Whether current is flowing in or whether it's flowing out on a cellular level is going to give you a different outcome. So there you can see the peak, and there you can see the direction. So this is these are the predictions that the model is making based on the simulation. This was the clinical trial that was run, not by us, that was run at UPenn, but which used the model. The question still remains, right? How, even though I've shown you this nice picture, and even though you've had this nice little clinical result, right? Why would putting a battery on your head, in this case, sort of make you read a little bit better? Right? It remains a sort of puzzling question. I do want to explain, I thought it would be important to talk about the difference between suprathreshold and subthreshold stimulation. So suprathreshold stimulation is like TMS. You basically give a shock and the cells fire. You give another shock, they fire again, right? You're increasing activity. You give 5 shocks, they fire 5 times. That's the basic idea. With subthreshold stimulation, which is like TDCS, when you apply stimulation, they don't fire. It's really weak. That 9-volt battery truly is not that powerful. And if your cells were sitting there and not doing anything, they're not going to respond. But your cells are doing something, right? Your cells, all the cells in your brain are constantly active, right? Your brain is really primed continuously. And in this situation, if you apply TDCS, you can interact with ongoing activity. You can modulate. And so again, I don't have the answer to your question, but I keep coming back to it. If in the obese head, the background activity or the background state of the system was different than in the healthy head, you would expect TDCS to affect these two things differently. That's a really hand-waving answer. So I did want to make that point. And so TMS, TDCS. Okay.